Welcome back, everyone. So please find your seats. And just to welcome you to this next session, Understanding and Restoring Alpha Cell Function in Type 1 Diabetes and Type 2 Diabetes, Intrinsic, Paracrine, and In Vivo Consequences in Humans. My name is Shareen Forbes. I'm Professor of Diabetic Medicine at the University of Edinburgh in Scotland and a clinician for the islet transplant program there. And we've got a great lineup of speakers. So our first speaker is Dr. Dale Morrison. Dr. Morrison is a research fellow and exercise physiologist at the Department of Medicine at the University of Melbourne in Australia. The main focus of his research is how physiology, nutrition, and exercise can improve the lives of those living with type 1 diabetes. And I'll let you introduce your talk.
Thank you. So we're going to have a bit of a change of pace after lunch today. I don't have a single picture of a stained islet or a single rodent. So hopefully I can keep you awake for the next 15 minutes or so. So I think given the nature of today's session, most people in the room will be familiar with the problem we're all talking about, which is hypoglycemia. And this remains a challenge for people living with type 1 diabetes today, despite the recent and excellent advance we've had in diabetes technology, such as CGM and automated insulin delivery. From my perspective as an exercise physiologist, hypoglycemia is especially problematic during exercise, where insulin requirements change rapidly and glucose disposal increases dramatically. And I'm also interested in the overnight period, which has historically been a concern around hypoglycemia for people living with type 1 diabetes. And I think we can all agree that additional strategies to manage hypoglycemia are still required in this population. And one of the key mechanisms underpinning hypoglycemia in type 1 diabetes, again, as I'm sure you're all aware, is the impaired or sometimes even completely absent glucagon response, which is seen in response to stimuli such as hypoglycemia and to exercise. And that's demonstrated by the couple of figures I have on this slide here. So on the, see if I can get this pointer to work. Not without looking, I can't, that's okay. On the leftmost slide, you can see that the usual counter-regulatory glucagon response in people without diabetes in the white squares is initiated at the beginning of exercise and increases to about 2 to 3-fold. And in the black circles, that's diminished in the people with type 1 diabetes. Similarly, on the right, in response to hypoglycemia, in this case a hypoglycemic clamp, you can see that as the blood glucose levels drop into the hypoglycemic range, again, there's a 2 to 3-fold and can potentially be an up to 8-fold increase in glucagon in the people without diabetes in the white squares and an impaired response in the people with type 1 diabetes in the black circles. And this is important as it limits the body's ability to increase endogenous glucose production to dropping glucose levels and to mitigate the resultant hypoglycemia. And so we're interested currently in looking at protein or amino acid ingestion in people with type 1 diabetes as a way to stimulate glucagon secretion and theoretically to help mitigate hypoglycemia in these people. And so the two studies I've cited here on the left are from the late 1960s to mid-1970s, which show that amino acids stimulate glucose secretion in humans, and that this stimulation of glucose secretion by amino acids remains intact in people with type 1 diabetes. And so this occurs despite the fact that there's an impaired glucagon counterregulation to exercise and hypo, as that appears to be a result of defective upstream paracrine signaling, which is absent or defective in type 1 diabetes, whereas the amino acids can directly stimulate glucagon secretion in a couple of ways, via direct transport into the alpha cell itself, and 17 of the 20 amino acids can stimulate glucagon via a variety of mechanisms in that way. And they can also indirectly stimulate glucagon secretion via the gut-derived incretins. And so theoretically, this should have a potent effect in stimulating endogenous glucose production in people with type 1 diabetes. And this can be demonstrated sort of in a summary figure on this slide, where as protein is ingested and passes through the GI tract, there's an increase in the circulating gut-derived incretin hormones, GIP and GLP-1, and a potent increase in the amount of amino acids circulating in the bloodstream. Together, these both stimulate glucagon secretion And in the absence of an endogenous insulin secretion, which occurs in type 1 diabetes, this results in a potent increase in the glucagon-insulin ratio in the portal vein, which is a very potent stimulus for increasing endogenous glucose production from the liver. Importantly, as you can see at the bottom of this slide, amino acids also act as a fuel for gluconeogenesis in the liver, and we know that glucagon upregulates the use of amino acids via gluconeogenesis, and theoretically all of this combined should work to increase blood glucose levels in people with type 1 diabetes. An important consideration, and one of the reasons why protein ingestion and amino acids maybe haven't received the attention they otherwise might have, is that the absorption rate and resultant peak amino acid concentration is what actually determines the physiological effect of the protein ingestion itself. And what I mean by this is the slowest absorbing proteins, your typical whole proteins like meat, eggs, fish, absorb quite slowly, result in a low peak amino acid concentration and therefore a low sort of glucoregulatory effect or a low glucagon secretory effect. Whereas the faster absorbing proteins such as, and we're looking at a bit later, whey protein isolate, absorbs really quickly, results in a high peak amino acid concentration and therefore a high glucoregulatory effect. And this can be demonstrated on the slide at the bottom, where the fastest absorbing proteins in the blue result in the highest peak amino acid, the highest peak glucagon, and therefore the greatest glycemic effect. And really importantly, one of the key things we're super interested in regards to protein ingestion and glycemia is whether, for lack of a better term, protein ingestion can awaken the sort of usual counter-regulatory glucagon response to hypoglycemia and to exercise. And I'll show you a couple of studies right now which explain what I mean by that. So this is a study from 2008 in people with type 1 diabetes where they induced hypoglycemia or ingested amino acids together or alone. And what we can see on this slide is that in response to hypoglycemia alone in the white circles, there was basically no glucagon response, as I discussed earlier, is to be expected in people with type 1 diabetes. Under euglycemic conditions with amino acid ingestion, there was a moderate increase in glucagon secretion, again to be expected in people with type 1 diabetes. But importantly, when amino acids were ingested prior to the induction of hypoglycemia, there appeared to be at the lowest blood glucose levels in the clamp, a restoration of some of that counter-regulatory glucagon response, which is usually seen in people without diabetes. Similarly, this is a study from earlier this year which showed a similar phenomenon in regards to exercise, where with people with type 1 diabetes, they ingested either a low protein or a high protein meal 90 minutes pre-exercise. And what we see in response to the low protein meal in the red is a modest increase in glucagon in response to the meal itself, again to be expected. But during exercise, there was no counter-regulatory glucagon response, which is usually seen in people without diabetes. On the other hand, in response to the high protein meal, again, there's a robust glucagon response to the meal, which is to be expected. But after induction of the exercise, there appears to be some sort of synergistic effect restoring that counter-regulatory glucagon response usually seen in people without diabetes. So despite over 50 years of evidence looking at protein and type 1 diabetes and glucagon, there's very little data looking at its potential utility for mitigating hypoglycemia. And so we aim to undertake a series of studies, firstly characterising the effect of protein ingestion in type 1 diabetes, and then using that data to inform some real-life interventions around exercise overnight and mitigating hypo. But today I'll just be presenting some data from our initial sort of physiological studies. So today I have data from 6 adults with type 1 diabetes who are using insulin pumps. Their characteristics are shown on the left. But importantly, they underwent 3 main interventions. So all participants underwent a controlled water ingestion, a low-dose protein, which was whey protein at a dose of 0.25 grams per kilo, and a high-dose protein intervention. And then 1/3 each of the participants underwent an exploratory intervention, as shown in red. So N of 2 underwent an extra high-dose protein, a carbohydrate, and a carbohydrate plus protein. And the nominal sort of protein doses based on the body weight are shown on the right. So the low dose was approximately 20 grams, high dose 40, and extra high dose 60. These doses were chosen based on sort of sports nutrition guidelines, which recommend 20 to 40 grams of protein pre or post exercise. And then the carbohydrate was approximately 20 grams, which is more or less similar to what people with type 1 diabetes use to treat their hypoglycemia. So our participants arrived at our lab, fasted overnight in the morning. They were all wearing insulin pumps, so we disconnected those and began an IV insulin infusion. We allowed a 4-hour washout period prior to the experiment to allow the subcutaneous insulin to wash out. And throughout this period, the insulin infusion was variable, but once we started taking samples, the insulin infusion was fixed. We also infused a glucose tracer, which allowed us to determine glucose turnover, so glucose production, glucose disposal. And then after ingestion of the intervention, we took blood samples every 10 minutes for 3 hours. Importantly, what did we see? So the next few slides I'll show all have the control condition in the black. Low dose protein is light blue, high dose medium blue, and extra high dose is dark blue. Carbohydrate red, carbohydrate plus protein is purple. And so this is the plasma glucagon response to the interventions. And I guess the very clear thing you can see immediately looking at this slide is that there's a clear dose effect of the protein ingestion on the glucagon response to protein. So the low dose peaked at about a 5-fold increase from baseline, the high dose about a 7-fold increase, and the extra high dose about a 9-fold increase. Importantly, the glucagon response was quite prolonged and quite sustained in regards to the protein, especially with the higher doses still elevated after 3 hours. And importantly, this is quite a substantial physiological effect. As I mentioned earlier, the usual sort of counter-regulatory response to exercise and to hypoglycemia is maybe a 2 to 3-fold and maybe even up to a 7 or 8-fold increase in glucagon. So these 5, 7 and 9-fold increases are quite substantial in that respect. This is our glucose appearance or endogenous glucose production data. Unfortunately, we only have an N of 1 for this because our mass spec broke down a couple of months ago, right as I was starting to analyse this. Essentially what this shows for the black, and so the control on the protein conditions is endogenous glucose production from the liver, and the carbohydrate condition is endogenous glucose production plus the ingested carbohydrate appearing into the system. And so what we can clearly see is in response to the carbohydrate, as you would expect, the glucose appears very quickly in the system, peaks very highly, but then after approximately 1 hour to 70 minutes, the amount of glucose appearing in the system via glucose production or appearance is back to baseline. So carbohydrate is excellent for treating imminent or current hypoglycemia. It works really quickly, it restores glucose levels, but doesn't have a very sustained effect. Whereas the protein appears to initiate an induction of endogenous glucose production within the first 10 minutes, which peaks after about half an hour, but is sustained importantly for several hours, and in the high dose condition, up to 3 hours. And we think that has really nice implications for protein ingestion to be used to prevent hypoglycemia over long periods of time, such as overnight, prolonged exercise or prolonged fasting. And it appears that the degree of stimulation of endogenous glucose production is more or less directly related to the extent of the glucagon secretion in response to the protein ingestion. This slide shows the glycemic response to the protein ingestion itself, again with control in the black and then the increasing sort of darkness of the blue, the higher protein doses, with this slide showing the change in blood glucose from time zero when the protein or when the intervention was ingested, millimoles on the left, milligrams per deciliter for those of you who work in those numbers on the right. And again, what we see is a very clear dose effect of the protein ingestion on blood glucose levels, with the low dose peaking at about 1 millimole greater than baseline, the high dose about 3 millimoles and the extra high dose about 4 millimoles, which may even be of a greater magnitude considering that the control condition, based on how long they'd been fasted, couldn't really reach a steady state. It was dropping throughout the entire trial as well. And importantly, again, the effect is very sustained, so that especially for the high doses, still potentially increasing or at least very high after the 3-hour analysis window has ended. And we know from previous data and some data I'll show you in a minute, that's even more sustained than this shows as well. When you compare it again to the carbohydrate, as you would expect, the carbohydrate has a more robust glycemic effect, which peaks after about 40 to 50 minutes at about 5 millimoles. But importantly, as we sort of touched on before with the endogenous glucose production, glucose appearance data, that effect is quite transient, and after the first hour starts to drop. And so implications for sort of preventing hypoglycemia over longer periods of time. And then again, as you might expect, the glucose plus protein condition in the purple is more robust than the same protein dose alone, but more sustained than the same glucose dose alone. And then finally, I have some pilot free living data that we've collected in adults with type 1 diabetes using insulin pumps in what we call manual mode or open loop mode, which basically means they had a fixed basal rate of insulin delivery overnight. And this was quite a simple study design. They had 2 separate nights where they either had a protein dose immediately before they went to bed or a water control immediately before they went to bed. And so this first figure shows data from N of 10 participants who ingested the low dose of protein compared to the water immediately before bed, which is time 0. And again, this slide is showing the delta or the change in their sensor glucose this time for the 6 hours post-ingestion. And again, clearly what we see is a substantial glucose effect, so about a 2 millimole increase from baseline for the protein condition, which turns out to be about 3 millimoles greater than the control condition, and an effect which is persistent across the entire 6-hour analysis window we used overnight. Similarly, for N of 5 participants who ingested the higher dose, which is 0.5 grams per kilo compared to the water, and the change in glucose, again overnight, was a little bit greater than the low dose alone, So again, peaking at about 2 millimoles, but about 3 to 4 millimoles greater than the control condition. And again, sustained up to 6 hours. So we think this has really promising utility for preventing hypoglycemia overnight in people with type 1 diabetes. In conclusion, we know that whey protein ingestion rapidly stimulates glucagon secretion to pretty significant physiological levels, which in turn stimulates glucose production from the liver and increases blood glucose in a dose-dependent and moderate way, which is more sustained than carbohydrate alone. And we think it's a really promising way to manage hypoglycemia in type 1 diabetes. Importantly, not only could it be effective, but it's cost-effective, well-tolerated, and should be easily translatable into clinical practice, given you can basically buy it at most supermarkets. And again, from an exercise physiology perspective, it's aligned with the sports nutrition guidelines. So a lot of people exercising potentially take protein already as part of their exercise regime. And so maybe just a small lifestyle change to sort of implement it in a more practical way. And then finally, in terms of sort of future directions, we want to tease apart some more of the physiological mechanisms regarding the effect of protein and glycemia. We have our first real-life sort of hypoglycemia mitigation studies starting later this month around exercise, but also overnight. And then the final point, which I touched on earlier as well, is whether we have some studies that we're looking at teasing apart whether protein can sort of restore some of that usual glucagon counter-regulation that's seen in people without diabetes as well. And so a big thank you to our team and our collaborators, specifically Sam, Zhang and Kara, who did a bulk of work on this, and obviously to the Helmsley Charitable Trust for funding this and for having me here today. It's been a true pleasure. Thank you.
So our next speaker is Dr. Zhang, who is an Associate Professor of Endocrine Physiology at the University of Oxford, and also an Assistant Researcher at the University of Coimbra in Portugal. Dr. Zhang will be speaking on hypoglycemia, sensitizes negative feedback control of glucagon secretion, underlies effective glucagon response in recurrent hypoglycemia.
Okay. Thank you very much for the introduction, and it's a great pleasure to be here. And I brought the Alice picture back now. I don't think I need to spend too much time on these slides, because we all know that pancreatitis is comprised of different endocrine cells. Beta cell secrete insulin, and alpha cells secrete glucagon, and prevent hypoglycemia, the main topic of today's symposium. And there's a third population of the delta cells. Sebastian also talked about it early this morning, that is secrete somatostatin. Somatostatin is a potent inhibitor of both insulin and glucagon secretion. Therefore, because of this role, it plays a very central role in the paracrine network within the islets. So we and others have shown before that delta cell, they can regulate beta cell in a reactive way. So there's negative feedback control. If a beta cell starts to be too active, it wakes up the neighboring delta cell, and delta cell then secrete somatostatin, and then will block a beta cell and to quiet it down. And this is very useful because it provides somatostatin on demand. And we wonder if this is some similar mechanism also work between alpha cell and delta cells. So the first question of our study is, do delta cell react to alpha cell activity? As we all know that glucagon secretion is regulated by glucose. At low glucose, there's a lot of glucagon secreted, and then it will be suppressed by high glucose. And the question would be, is somatostatin also secreted at a low glucose too? To answer this question, we generate a transgenic mouse model. So we knock in G-campus, the calcium probe, into the delta cells alone. And then we can monitor delta cell activity using calcium imaging technique. So here, this video shows the delta cell activity. At low glucose, at 1 millimolar glucose. As you can see, those sparks, green spark flashes that shows the delta cells are active. They have a higher intracellular calcium concentration. So approximately 38% of the delta cell actually are active at 1 millimolar glucose. So So delta cells are active at low glucose, but do they affect alpha cell and glucagon secretion? To answer this question, we use hormone secretion experiments, and we measure glucagon secretion in response to CYN, that is the somatostatin receptor blocker. And you can see here, it is strong. It is pretty strong effect. It has pretty strong effect at all different glucose concentration, but most strikingly, the effect of somatostatin receptor blocker is really strong at low glucose, much higher than at high glucose. So this indicates that delta cells have a stronger control over alpha cell glucagon secretion at low glucose. How does it work then? I mean, could it be simply that because somatostatin secretion is also high at low glucose? But as it has been tested many times, we also got exactly the same result. At low glucose, somatostatin secretion is actually very low. It's about less than 10% of what you get from high glucose. So it seems, so the question will be, how does this low level of somatostatin can regulate alpha cells so potently at low glucose? So we have a hypothesis. We think the alpha cell and delta cell, they are tightly coupled together. So whenever alpha cell a little bit too active, so this would, again, wakes up the neighboring delta cell, and the delta cell would give a feedback regulation right to the alpha cell to prevent it from over-secreting. So this would be cell-to-cell resolution, and it will be very fast, and depends on alpha cell activity. So in this way, you don't need so much somatostatin. Already you can efficiently regulate alpha cell at low glucose. So to prove this, to test this hypothesis, we generate another mouse model using optogenetic technique. So we knocked in this time a light-sensitive channel into the alpha cells. So this, when we give a light to the iris, we can selectively turn on the alpha cell using remote control the alpha cell. So we switch on the light on, the alpha cell should start to do some secretion. And that is exactly what we saw the result. When you switch the light on, glucagon started to secrete much more, and so the model works. But what is interesting is that somatostatin secretion in this iris also responded to the light stimulation. And I just want to remind you that the channel rhodopsin, that the receiver of the remote control, is only expressed in alpha cells, not in the delta cells. Therefore, so we can conclude from here that the controlled stimulation of alpha cell activity actually stimulates somatostatin secretion. And we wanted to further demonstrate this interaction. So we look at a single cell resolution. So we use the GCAMP iris, the iris that express GCAMP in delta cells, and we can patch a neighboring alpha cell, and we give a stimulation to the alpha cell, and we can monitor the delta cell response to the alpha cell activity. And if you put that into action, so the sound there marks the stimulation onset of the alpha cell. So this cell, not this cell, but I guess you also, there was a flash in the delta cell. So delta cell did answer to the alpha cell activation. And this is very fast delay, about 700 milliseconds. This interaction is dependent on the alpha cell excitosis in a dose-dependent manner. So the more excitosis happening during this process, so the red line shows here, when we increase the strength of stimulation to the alpha cell, you can see the delta cell started to become more and more responsive. And if you plot everything together, it's quite nice positive correlation between these two different parameters. So there is interaction, and they do talk to each other, but what is the mediator? Alpha cells, they secrete not only glucagon, but also glutamate. Glutamate is a neurotransmitter that can activate ionotropic glutamate receptors. And delta cells, they actually are expressing the glutamate receptors too. So it's a perfect match. then we test the role of glutamate in the interaction between these two different types of cells, and we can see that if you block the glutamate receptor using these two different blockers, AMPA receptor blockers, and you can reduce the somatostatin secretion at low glucose. And if you use glutamate, it strongly stimulated somatostatin secretion at low glucose. And at a single cell level, using the exact the experimental setup I showed earlier, under control condition, you gave a stimulation to alpha cell, delta cell did answer. But when you have the presence of the AMPA receptor blocker, delta cell become insensitive to the alpha cell activity. So it shows quite nicely that there is an interaction between these two different cells, and this glutamate is the mediator. So why do we think this is important for hypoglycemia? So when you treat diabetic patient, there's a clinical studies look at patients with diabetes, and if you see you have one episode of a hypoglycemia, it's very likely you will have another one very soon. So it's a hypoglycemia begets hypoglycemia, and the reason being that if you have one hypoglycemia, this is a study that is done by Philip Creer in 1991. So if you induce two sequential hypoglycemia on two separate days, the glucagon response to hypoglycemia on the second day is much weaker. So it seems hypoglycemia can form a kind of long-lasting metabolic memory. So because we found this new interaction, we wonder if that is true, the alpha-delta cell interaction. So we want to test this idea. We first modeled recurrent hypoglycemia in mice. So this is a 2-day protocol. On the first day, we give mice an injection, either saline or insulin. So insulin does exactly what you expect it to do. It induced hypoglycemia, and saline didn't do much. And on the second day, if we do an insulin tolerance test to check to test the counter-regulation in the mice, and you can see these mice that had hypoglycemia the day before. It has much deeper and longer hypoglycemia on the second day. And this probably is because their glucagon secretion is no longer stimulated by low glucose. So there is a metabolic memory on glucagon secretion. Could this be due to alpha-delta cell crosstalk? And if we test this by blocking the somatostatin receptor on the second day of the experiment, as you can see here, adenine C1N completely restored counter-regulation in the mice, and the glucagon is also back. So all good story. And then we can test this in the in vitro setting. So we we have this transient exposure of low glucose of the islets, and then we recover them for overnight in normal glucose just to mimic the in vivo situation. And you can see using the same patch imaging technique as I showed earlier that if in the islets that had experienced a hypoglycemia the day before, the alpha cell, when you stimulate the alpha cell, delta cell responded in a much faster and stronger way. So there is a sensitization of this interaction. So how does this interaction got sensitized? We have another hypothesis. We think that during the first episode of hypoglycemia, there's a lot of glucagon secreted, and then the glucagon binds to the glucagon receptor in the delta cell, and that potentiates the delta cells. So the delta cells become much more sensitive when the next episode of hypoglycemia hits. So in this way, it locks down alpha cells. So the delta cell become too jumpy. So whenever there is a little bit of action coming from the alpha cells, delta cells started to secrete somatostatin to lock it down. So this can cause a vicious circle of recurrent hypoglycemia. So we test this hypothesis using a different mouse model, this time using a chemogenetic mouse model. So it's the same principle as the optogenetic mouse model, also using a remote control system. So this time we use a chemical to regulate alpha cell activity. In these mice, so if you inject artificial ligand, the G60, into the mouse, and the alpha cell specific, we can turn on alpha cell specifically in vivo. So on the day 1, we gave injection to activate alpha cells in the mouse under normal glucose condition. So there's a control mouse that expresses the protein, and the dread mouse expressed the protein, and the control mouse that didn't. And you can see that when we give the agonist, the mouse responded nicely with increase in glucagon, and there's no hypoglycemia whatsoever in the mouse. And in the control, there was no response at all. And on day 2, these mice that experienced hyperglucanemia instead of hypoglycemia, they phenocopied exactly what we saw in mice exposed to hypoglycemia before. So there's much longer and deeper hypoglycemia, and there's a lack of response, glucagon response to hypoglycemia on the second day of the experiments. So glucagon is the factor that shapes the metabolic memory between alpha and delta cell within the islets. So I would like to conclude my talk using this slides. That this is what we normally see in the islets, that alpha cell and delta cell, they interact. So alpha cell, whenever there is activity, when it reaches certain threshold, can activate delta cells, and delta cell feedback with somatostatin just to make sure it's not over-secreting. But when in a pathophysiological condition, when there was one episode of hypoglycemia, the sensitized this interaction, then it can become like this. So it's just a little bit of alpha cell activity is enough to trigger the delta cell response to clamp down the alpha cell activity. So if we can understand what is the mechanism behind this metabolic memory, and if we can restore the normal alpha-delta cell interaction, we should be able to prevent recurrent hypoglycemia. That's the hope. And finally, I would like to thank the people that are involved in this study. Patrick has been a long-term mentor and a collaborator, and redid most of the experiments beautifully. And I would like to thank the collaborators from Shandong University, University of Alberta, and Davis Group, and also Nanjing University, and also my founders. Thank you very much.
So it's a real pleasure to introduce our next speaker, Dr. Michael Rickels, who is a professor in diabetes and metabolic diseases at the University of Pennsylvania, Perelman School of Medicine, and director of the Clinical Science and Endocrinology and Diabetes program there, and also the medical director for the islet transplant program there. Michael's really led the way in our understanding of many of the metabolic scenarios in islet transplantation, and I'm delighted that he's giving the talk today, Understanding and Restoring Alpha Cell Function in Type 1 Diabetes. Thank you.
Thank you, Shareen, for that introduction, and to Ben and Kanchi and members of the organizing committee for the invitation to join you today for this fantastic symposium. I've learned so much already. I hope I can add a little bit more to the great lessons already shared today. So as a bit of a review from this morning, there's clearly impaired alpha cell function and gene expression that's been shown in type 1 diabetes. And looking at data from the Human Pancreas Analysis Program on the left, I want to point out that what happens in the progression of type 1 diabetes is that as the beta cells are lost, it appears that that's beta cells being lost from entire islets, and that when there is residual beta cell function, that's from lobules within the pancreas that have seemingly normal intact islets. And so you can see that on the image in the middle, where the immunohistochemistry shows this is from an individual within 2 years of diagnosis of type 1 diabetes. And you can see a lobule that has entirely normal appearing islets, and in the rest of the pancreas are islets devoid of beta cells. And at the bottom, you can see an individual here with 5 years of diabetes duration who was found to have residual beta cell function. And here, they occur in islets within lobules where you can see the islets in the middle have no beta cells present, but the islets surrounding them have a pure normal complement of beta cells. And so what I think this means is that the residual beta cell function is coming from residual intact islets, but the vast majority of the glucagon that's coming from the pancreas in someone with type 1 diabetes is coming from islets that don't have beta cells. And from this case, when you can pull the parafusion data from this case off of the PancDB website, and you can see that the insulin secretion from islets isolated from that pancreas quantitatively is very low, but qualitatively, the insulin secretion responds appropriately to increases in glucose, whereas the glucagon secretion from islets isolated from the same pancreas fails to suppress glucagon secretion in response to high glucose. And so, as Marcela Bersova showed so elegantly in this paper that Lori highlighted this morning, the alpha cells in the isolated from these donors with type 1 diabetes have normal glucagon content, but the regulation of the identity and function of those alpha cells is impaired. And so what does that mean for the paracrine regulation of islet function in type 1 diabetes? So we've heard lots about this paracrine regulation so far today. And of course, glucose is central to all of this because it stimulates the beta cell while suppressing the alpha cell. And so as you remove glucose from the situation, as hypoglycemia develops, you take away stimulation of the beta cell, which then, as the beta cell turns off, that takes away the brake on the alpha cell as well as directly as well as indirectly through the delta cell, and allows for the increase in glucagon secretion that's required to prevent further lowering of the plasma glucose concentration. But in type 1 diabetes, the loss of paracrine regulation seems to be largely due to just the islet being devoid of beta cells. And so there's no beta cells present in the islet. And as you've heard already, there's this tonic increase in delta cell activity that keeps a brake on the alpha cell. And then we also have to remember that we're treating our patients with exogenous insulin that is peripherally administered, and so requires higher systemic levels to reach portal concentrations that control liver production of glucose. And this exogenous insulin also has its own inhibitory effect on alpha cell function. And then, of course, we've also learned that when mealtime occurs, amino acids can stimulate the alpha cell, and that occurs sort of despite where the glucose concentration is, as does GLP-2. GIP is the one that has more of a glucose-dependent effect on alpha cell function, so that is stimulating glucagon secretion at low and not high glucose, whereas you've seen already amino acids as well as GLP-2 can stimulate the alpha cell at high glucose. But under conditions of hypoglycemia occurring in the interprandial periods, there seems to be no ability to counteract the effect of exogenous insulin and increase glucagon. So how the beta cell does that communication, I want to just point out the role of insulin as one of those factors, because that's what we use therapeutically. And here, Ben Cooperberg, when he was in Phil Cryer's lab, showed this in an individual with type 1 diabetes by infusing a really high amount of insulin to sort of simulate the high intra-islet insulin concentrations you might imagine being present, and then doing that intravenously so you could then turn off insulin at 60 minutes and let the insulin concentrations drop. And doing that or sustaining the high insulin under conditions of either maintaining euglycemia or allowing hypoglycemia to develop shows that in these individuals with type 1 diabetes, under insulin turned off and low glucose, you could get a modest increase in glucagon secretion relative to the conditions of sustained high insulin or sustained euglycemia, suggesting that these alpha cells do have some modest ability to respond to this high intra-islet decrement of insulin secretion. And so what we know about this normal response of the islet cell to lowering plasma glucose, that the decrease in insulin secretion, and whether some of that is directly caused by insulin or from direct communication, as we learned earlier, other paracrine mediators, that decrease in insulin is obligate for an increase of glucagon secretion in order to increase hepatic glucose production and restore plasma glucose. And so with beta cell loss in type 1 diabetes, we lose not only the ability to remove insulin at the time of hypoglycemia, but also the ability to increase glucagon secretion from the alpha cell. And so hepatic glucose production cannot be increased, and defective glucose counterregulation results. And this glucagon response has been shown by Anna Maria Arbolez at WashU to already be markedly impaired at diabetes onset. These are data from adolescents at diagnosis, and then after a year of achieving glucose control. And during the same hypoglycemic clamp, you can see relative to controls that at diagnosis and following a year later that the glucagon response is markedly impaired. And what these patients do have at diagnosis is an intact epinephrine response to defend against hypoglycemia. And we were interested in looking at the role of residual beta cell function on that alpha cell response. And here are data looking at nutrient ingestion. And so we did this a study as part of the type 1 diabetes exchange and recruited individuals with type 1 diabetes who had what we considered a high amount of residual beta cell function or an intermediate amount, which was above the DCCT threshold, or a detectable but low level, as well as those with no detectable beta cell function. And you can see, while there's clearly distinct beta cell responses during a mixed meal tolerance test, that the effect of amino acids, gut hormones on the glucagon response during that meal was the same regardless of how much residual C-peptide. So, and my interpretation is that you're only going to get suppression of glucagon within the islets from those few remaining intact islets that are left in the pancreas, but the overwhelming number of islets are going to have lost that paracrine regulation and so are stimulated similarly. And you see the same thing with glucose-potentiated arginine, where with a glucose infusion and then administration of arginine, you see a progressive increase in the amount of the beta cell response, but despite that, no difference in the regulation of the alpha cell response to release glucagon to arginine, regardless of how much residual beta cell function. Again, because I think we're measuring the glucagon from the vast majority of islets that have lost paracrine regulation. Where you can see it is in the alpha cell response to hypoglycemia. So here we have the same individuals with beta cell responses of high, intermediate, low, or negative, and they all had a hyperinsulinemic U followed by hypoglycemic clamp. And so you can see clear differences in the beta cell turnoff. So going from U to hypo, we see this graded magnitude of the reduction of the beta cell turnoff. And so this is only occurring in those intact islets. And what you see is when we measure glucagon, high insulin suppresses glucagon, so there's a suppression into euglycemia. And the increase of glucagon during hypoglycemia was only significantly greater in those with the highest amount of residual beta cell function. With the other groups, I believe because they just don't have a sufficient number of intact islets release glucagon to differentiate from the group that is C-peptide negative. So it takes a high amount of residual beta cell function to maintain even a modest glucagon response to defend against hypoglycemia. And so very interesting work, and Asker Lung is going to talk about this later, I hope. So GIP has been shown to augment the glucagon response to hypoglycemia in type 1 diabetes. And so in this study, hypoglycemic clamp was done on the, shown on the left. And what they did here, the high insulin that was used to induce hypoglycemia at 60 minutes was turned off. And so you see in the middle, you see the insulin concentrations going down. And again, you can achieve this when you give insulin IV. This is not what happens when insulin is given subcutaneously. And then you can see that relative to the saline control or GLP-1 infusion, which has inhibitory effect on the alpha cell on the right, you can see that the GIP infusion led to an increase in glucagon. And so this does appear to be glucose dependent in the activation of the alpha cell, but it also wonder how necessary that turnoff of insulin was to enable the alpha cell to respond. That is, what would happen in the context of ongoing hyperinsulinemia. And so there was a 6-day subcutaneous infusion trial of GIP that did not reduce time below range relative to placebo in individuals in type 1 diabetes. So I think that is one of the remaining questions to answer, is how sensitive is this effect in the presence of ongoing hyperinsulinemia. There's a GIP hypo study that's now aiming to determine optimal dose and isoforms of GIP for the alpha cell response to hypoglycemia in individuals with type 1 diabetes, and I hope Asker might share some insights on this later. So to restore the paracrine regulation of islet function, we might also think outside the islet to agents like GIP or other ligands of G protein coupled receptors that might work to stimulate an increase of glucagon. And the question still is, can that override the inhibitory influence of the exogenous insulin present at the time of hypoglycemia in our patients? Another approach, and Richard Liggins is going to talk about this as well later this afternoon, is blocking somatostatin with somatostatin receptor 2 antagonists, ZTO1. And so can this interfere with this tonic inhibition of the alpha cell by the delta cell and again allow for a glucagon response to occur? And so Dr. Abittal presented at this conference last year that ZTO1 increases glucagon during a U hypoglycemic clamp. The way this clamp was conducted was with a variable insulin infusion, and then baseline samples were collected at period A, and then under euglycemic conditions B, and then at 2 steps of hypoglycemia C and D. But what this entailed was following a treatment at A, there was increasing delivery of insulin so that the insulin exposure varied over the periods. There was a response of the alpha cell to increase glucagon that was already present at period B under euglycemic conditions, did not appear to increase further during hypoglycemic conditions, but there was also a further exposure to insulin during this period. So again, looking at the separating the glucose dependency from the effect of changing insulin, I think is going to be an interesting question to address. And a study of the effect of ZTO1 on nighttime hypoglycemia type 1 diabetes is currently ongoing, and I think we may hear more about that and other plans from Richard this afternoon. So there's also this autonomic, there is autonomic innervation and regulation of the pancreatic islet, and so this regulatory system that occurs as well as the paracrine system we've been focusing on. And so the activation of the parasympathetic nervous system stimulates both the beta and alpha cell secretion, activation of the sympathetic nervous system, inhibits the beta cell through alpha adrenergic receptors, and activates the alpha cell through beta adrenergic receptors. If that's not enough alphabet soup for Lori, so how important is this to the problem of the glucagon response in type 1 diabetes? And one thing I think is important to appreciate is that there's long been a dissociation of the glucagon response from the epinephrine response in type 1 diabetes. And so I'm just sharing an example from data from our lab where we use stable glucose isotope to measure endogenous glucose production, fixed hyperinsulinemia, and then variable glucose to have either a euglycemic control clamp or a stepped hypoglycemic clamp. And from several cohorts we've studied, you can see on the bottom, the gray shade gives the data during a euglycemic clamp, and you can see that the cohorts with impaired awareness of hypoglycemia or a group with intact awareness of hypoglycemia have absolutely no glucagon response to hypoglycemia, with levels not different than under the euglycemic control conditions, and that's relative to a non-diabetic control group that has a normal glucagon response. But when we look at epinephrine, we see that relative to euglycemia, those with impaired awareness of hypoglycemia do have a very modest epinephrine response. Those with intact awareness, these are now duration matched. So by this point in the disease, unlike Anna Maria's slides I showed earlier, their epinephrine response is no longer normal, but it is different from those with impaired awareness, but yet there's no difference in the glucagon response. So this suggests that there's no effective epinephrine acting on the alpha cell during hypoglycemia. And that's largely because this is during insulin infusion. And so recent work from Dr. Shea is demonstrated with fixed dose insulin infusion, variable glucose to maintain euglycemia, but then giving a graded epinephrine infusion, that the glucagon response, whether it's a non-diabetic individual or someone with type 1 diabetes, remains entirely suppressed. So under high insulin consistent with postprandial insulin levels, epinephrine won't stimulate the alpha cell to release glucagon. So one of the areas we've been interested in is replacing alpha cell function through islet transplantation. And so this is typically done for an individual such as whose CGM tracing is shown here with quite labile glycemia and lots of exposure to hypoglycemia. Time in range is only spent when moving from hyper to hypoglycemia. And currently, this is done through the procurement of a deceased donor pancreas from an individual without diabetes that's taken to a CGMP facility for collagenase digestion, centrifuge purification. With the islets, it can be returned minimally invasively through a percutaneous transhepatic access to the portal vein. This is the resulting controlled glucose homeostasis, which can be normalized in individuals following receipt of islet transplantation. And now there's a lot of progress has been made in the development of human pluripotent stem cell-derived islets that have entered phase 1/2 clinical trials as an alternative source of cells than deceased donors. We showed almost 20 years ago now, when we did our first islet transplants following the Edmonton protocol, that during a hypoglycemic clamp, you could see the beta cell response. If you had a group with type 1 diabetes, there's no C-peptide, so there's nothing to turn off. Whereas those that received an islet transplant, they turned off insulin secretion from their intrahepatically transplanted islets, the same as our normal controls. And with that, we can see that insulin suppressed glucagon secretion in type 1 that was higher in the group who'd received islet transplants, but clearly not normal. And that's where the euglycemic control experiments are important to show that under euglycemic conditions, high insulin suppress glucagon secretion from the islet transplant individuals, the same as non-diabetic individuals, but on their hypoglycemic clamp condition, there was a clear increase in glucagon. Now, the group in Germany showed similarly that individuals with or so-called partial islet grafts in the liver that don't restore insulin independence, but provide a beta cell response that does turn off during hypoglycemia and allows for a modest increase in glucagon during hypoglycemia that nonetheless provides significant impact on liver glucose production that is likely what helps even those islet transplant recipients dependent on some insulin to avoid further hypoglycemia. And with the Clinical Islet Transplant Consortium protocol, where we had in this cohort of 10 individuals, all who achieved insulin independence, saw that exposure to hypoglycemia was essentially abolished. And here you can see, studying now the same individuals from before transplant to following transplant, that the glucagon response is about 40 to 50% of normal already at 6 months and stays there when studied again 18 months later. And here again, you could see the epinephrine response, which improved with hypoglycemic avoidance at 6 months, was further improved at 18 months, but without any difference in what happened to the alpha cell response. And then lastly, that the hepatic glucose production seemed to be entirely driven by what we saw with the glucagon response, already restored after 6 months without really any further change at 18 months, despite the further increase in epinephrine. And when we've studied a similar cohort with hybrid closed loop intervention for hypoglycemic avoidance, we got people under the 4% target, but no hypoglycemia. And of course, as we've learned, that this will do nothing for the glucagon response to hypoglycemia in these individuals. And while it did modestly improve their epinephrine response, without glucagon, there was no restoration of the endogenous glucose production response. So again, highlighting the key role that glucagon plays in the defense against hypoglycemia. And so we wanted to look at the assessment, we wanted to assess the adrenergic contribution to this counter-regulatory response, and did that in our cohort of islet transplant recipients, where we hypothesized that alpha adrenergic blockade would block the decrement of insulin and hence paracrine signaling to the alpha cell to increase glucagon secretion. And that also that beta adrenergic blockade, if it was important for the alpha cell response, would similarly prevent glucagon secretion and increase endogenous glucose production. And so we tested this using a tracer-enhanced hyperinsulinemic U followed by a hypoglycemic clamp. We used a stable glucose isotope, fixed insulin infusion, and then glucose we met a target within 90 minutes of 80 to 90 milligrams per deciliter, and then by 180 minutes went to 45 to 50 milligrams per deciliter. And individuals came in on 3 occasions with double-blind administration of the alpha adrenergic antagonist phentolamine or the beta adrenergic antagonist propranolol or placebo in a crossover design. And for this study, we had 9 participants from the Clinical Islet Transplant Consortium protocol for islet alone transplantation. 6 of those came from Penn, and 3 were recruited from the University of Minnesota. And you can see their characteristics on the left. They were median 7 years following islet transplant with A1C median 5.8. 5 were insulin independent still. 4 were had returned to some insulin use, but were on low-dose insulin with a good islet graft function. You could see their CGM data, which is 95% in range and still with no exposure to hypoglycemia. And so when we did our experiments, this is our condition of hyperinsulinemia, so about postprandial insulin levels that are achieved, and then glucose that was matched at baseline as well as during the euglycemic phase and the hypoglycemic phase of the experiments. And then I'm not showing the blood pressure data with that was lower with phentolamine. Here's the heart rate data, lower with propranolol, that just shows, clearly how the beta-adrenergic blockade prevents the normal increase in heart rate you see during the induction of hypoglycemia. And so, first off, we saw no effect of either the alpha or beta-adrenergic blockade on the intrahepatically transplanted islet cell responses to hypoglycemia. The beta cell response was similarly suppressed under U by hyperinsulinemia and then further under hypoglycemic conditions, whereas the alpha cell response suppressed under euglycemic conditions by hyperinsulinemia, but similarly increased. In fact, the response during propranolol infusion was actually greater than it was under placebo, arguing against a role of epinephrine being important. We could see the beta receptors were blocked, again, not only by heart rate, but also with failed inhibition of epinephrine's own secretion. The epi response was actually higher in the face of propranolol. And then importantly, there was no difference seen between conditions on the hepatic glucose production, supporting the primacy, again, of glucagon over epinephrine for the counter-regulatory defense against hypoglycemia. So to summarize, alpha cell dysfunction in type 1 diabetes results from loss of beta cells within islets and ensuing paracrine dysregulation that leaves the alpha cell blind to hypoglycemia. Recovery of the alpha cell response to hypoglycemia from islets devoid of beta cells is going to require a solution that is both glucose dependent, and we want this to work at low and not at high glucose, but also that has to overcome the inhibitory influence of hyperinsulinemia that's present when our patients are developing hypoglycemia. Islets replaced in type 1 diabetes by transplantation to the intrahepatic site appropriately respond to hypoglycemia with beta cells turning off and alpha cells turning on, resulting in restored glucose counterregulation and defense against hypoglycemia. And physiologic alpha and beta-adrenergic blockade has no effect on that transplanted islet cell response, supporting, again, the importance of islet glucose sensing and paracrine rather than neural regulation for the alpha cell response clinically. With that, I'd just like to thank the members of my lab and collaborators at Penn who contributed to the work I presented, as well as our collaborators from University of Minnesota who referred patients for our adrenergic study, and to the support we've received for this work from the NIH, JDRF, now Breakthrough T1D, W.W. Smith and Helmsley Charitable Trusts, and colleagues in the Type 1 Diabetes Exchange study. And thank you for your attention.
With that, I'd like to thank you, Michael, and all the speakers for some really excellent talks, and for the audience for their questions as well. Thank you.